right. So there'll be a couple of slides in the beginning that are going to come from the last block, but anything from block 1 that they can be accountable for in block 2 will make a presence in block 2's slides. I'm not going to draw from anything that isn't in this block slide, so you don't need to re-watch old stuff, but there will be some re-emergence of some older content. But today and next lecture, we're going to talk about genes and genetic expression. We've been talking about hypertrophy and what is hypertrophy, but protein synthesis outpacing degradation. And what is protein synthesis? It's gene expression. So this is the next step after mTOR, is what is translation? mTOR is turning on translation. Hypertrophy, again, is anabolism, hypertrophy. These are protein synthesis happening at a faster rate than the degradation of protein. And so you get protein accretion, you get accumulation of protein. And turning on gene expression in hypertrophic ways is hypertrophy. That's anabolism. And it's also, to talk about this is a nice rational follow-up to mTOR. But we can also talk about, at the very end of next lecture, we'll transition from genetics into geriatrics, going from gene expression into what does senescence look like? What does aging look like? How is aging manifested in a genome? And we've been talking about a lot of the problems of aging, which would be, increased adiposity, lowering of skeletal muscle mass, bone density, stuff like that. But the cognitive effects, that's really what scares people, and that's what characterizes senescence, aging, this atrophy and deterioration of the human form with each passing decade and passing year, and at some point each passing week practically becomes noticeable. And there's a nice transition going from genes into aging. And then we'll talk about aging as a quick transition into mental health, cognitive health, brain health, and exercise science. The scores, where to find everything. So again, as we continue to do case studies, just remember the ACSM stuff. This is foundational to day 1, minute 1, an encounter. Maybe minute 1 is a greeting. Minute 1 is let's try to be charming so that we secure a patient or client or something. Minute 2 is let's figure out a risk profile of somebody, no matter what the goal is. If they're an athlete hoping to be a better athlete, if they're an injured athlete hoping to get back on a court or a field or whatever, if they are an injured non-athlete and they're hoping to regain function, if they are hoping to prolong life or optimize some components of health and fitness, you always begin with this risk stratification, and then within that risk stratification, figuring out how many risk factors there are. So categorize, stratify a person, and then within those strata, within those categories, figure out the number of risk factors. And we will talk more about metabolism in this section. We'll do a little spot on glycolysis. We've only briefly mentioned glycolysis before. We'll have one day probably on glycolysis. But metabolism, as we talk about BDNF, brain-derived neurotrophic factor, and we talk about brain health, that's metabolic also. So at the foundation of a lot of physiology is metabolism. And we have briefly mentioned glycolysis. I think it was lecture 4, lecture 5, something like that. There was this slide. All I did was add you've got glycolysis. You guys have all seen Lord of the Rings. I don't know if that's in The Hobbit or Lord of the Rings. Nasty little pockets is that line. So glycolysis, we will have a day where we talk about glycolysis because most understanding of it is a cheapening of what it does, how it works, and its consequence. There's sort of a crude cheapening of how we talk about glycolysis. And to get the real story, we'll go through a few of those steps, a few of those major steps. Now, with mTOR, you don't need to know the whole thing anymore. You don't need to know all of mTOR, but remember the basics of mTOR signaling because you'll see it in neurons. You'll see it in all of these cells. And not to discard it or forget it completely. We have the amino acid, nutritional, let's localize mTOR to the lysosome. Then we have the growth factor activation going through RAG. So just get RAG and RED, GTP loaded, and we have translation on. Hypertrophy is what we were talking about, but translation gets turned on. And then remember AMPK is the opposite of mTOR signaling. Over here we have translation, which is being inhibited by AMPK. Or AMPK inhibits translation and activates degradation. And so mTOR signaling, MAPK and PKB-mTOR signaling are activating translation. AMPK is deactivating translation. And moving into genetics and genetic expression and what translation is, we have a relationship between our environment and our body. So think what fitness is in general. A very general definition of fitness is your suitability to your environment, your appropriateness to your environment, your compatibility with your environment. That's what fitness means, is your body, your cells, your tissues, your organs, your whole whatever spirit is compatible with life's stressors, life's loads, right? Life's presentation of stimuli, that your body is compatible with that. That's what fitness is. And our genes are a great way of trying to optimize fitness. Tons of work on this, talking about the interplay between our DNA, what you're born with, your genes, your genome, what you're born with, and your phenotype, the body that you become. So your genotype and your phenotype. We'll talk about those things today. There's 1,000 cliches about this. A good one, but it's still really stupid because I've heard it conservatively. I've heard this one 500 times. And every single person who says it thinks that they're coming up with it. But it's in like 100 articles and 100 books, and it's been expelled from 1,000 mouths every minute, is that your genetics load the gun and the environment pulls the trigger, right? You need both of these things. Typically, you need both of these things. You need the genetics and you need the environment, and there's an interplay between them. Concepts of determinism are in part determined by the environment in which you grow up. Now, Nostradamus, this is just me talking, but here's a picture of Nostradamus and me putting words into his mouth, which is, hi, I'm Nostradamus. For the foreseeable future, everyone knows who Nostradamus is, right? He was a reasonable apothecary, sort of a medical guy. Apothecarial sciences weren't very favorable in his day. So he's actually expelled from school for doing this manual labor apothecary stuff. But plague era, he was reasonable. He was a reasonable kind of physician figure during the plague. And then he got a lot of confidence and wrote a bunch of predictions. And the predictions were so vague that they mean everything. And so anything that has ever happened in the future, Nostradamus predicted that. I guess. I mean, he's predicted everything ever with his just ambiguity. Anyway, for the foreseeable future, the only reliable opportunity we have to reduce disease risk in genetically susceptible folks, and the most reliable way to change other health parameters, is not the re-engineering of our genes, but modifying our environments. Again, he didn't say this, whatever. I'm just putting words in his mouth as a sort of prediction. Maybe in the future, CRISPR and whatever will be employable. Maybe at some distant date, we'll be able to engineer our genomes for more targeted therapies to address everything from disease and decay to the optimization of health and human performance, whatever. Maybe, sure, probably, but not now and not soon. In the foreseeable future, what we can do is change the presentation of stresses and stimuli and loads, change environmental contexts, and our body will respond accordingly to optimize fitness, to optimize compatibility to our environment. You should recognize this slide. Not everybody is going to behave the same way to the same presentation of stresses, to the same presentation of load profiles, of exercise, stimuli and stress, right? So some people will go do a cardiovascular, everyone does the exact same cardiovascular exercise, let's say. Take everyone in this room and we all go do 30 minutes on the treadmill and whatever, some exact percentage of our VO2 max, same conditions, our diet, everything is identical, and we respond differently. Some respond better than others, some simply respond differently from your peers, but some people respond badly to these things. Remember your HDL, you want that to go up, and you do see people going up in HDLs, but you also see this, HDL is going down with aerobic exercise. Systolic blood pressure, you want that to go down in most people. If you are hypotensive, that's not what you want. But in some people, you see an increase, this chronic increase in blood pressure. Fasting insulin, plasma triglycerides. We want these things controlled, and exercise in most people, in most circumstances, does a good job at controlling these things, but not everybody in every circumstance. And this is a genetic susceptibility. There are genomes that predispose us to being responders, non-responders, or poor responders, sort of inverse responses, where we're getting deterioration or we're increasing risk. It's not a high percentage of people, but there is genetically a percentage of people who respond badly to behaviors we consider healthful. So when we exercise or when we go up a mountain, right, go hiking, go biking, right, go to the weight room, whatever you're doing, you are changing protein content, you're changing genetic expression, and not everyone changes the same. So the importance of proteins in the body, this could be the entire class, first lecture through the final. You know, let's begin on word 1 of the syllabus and the last word of the final, and we talk about nothing but the value of protein. Could totally do that, but we're going to do a really cheap version of that right here, which is here are a bunch of categories of proteins in the body. Just kind of blunt, crude categories of proteins. You know, hormones and binding proteins. Down here, this is hemoglobin, you know, the oxygen carrying protein in red blood cells. Now, when you look at a box of cereal, like Cheerios or something like that, the Cheerios themselves are huge. You'd be able to get like 9 of them in a bowl or something, but real life Cheerios are kind of small. Hemoglobin, you're gonna have, I mean, just like, look how huge it is relative to the red blood cell. There's gonna be 250 million in there, maybe 270 million. There's tons of these things. These proteins are tiny. These are tiny proteins. But hemoglobin is a protein, and that's how you're alive and transporting oxygen. That's one of tons of proteins that permits your survival, right? It's hemoglobin. But hormones and binding proteins, we could put cytokines up there in that same category, but that's already getting kind of long. So hormones, not steroid hormones, right? Made from cholesterol. Steroid hormones is a little bit different, but most of your hormones, it's a bunch of amino acids, right? Sometimes it's an amine hormone. It's one amino acid. Take tyrosine, modify it a little bit, and now we have, we have dopamine, we have epinephrine, whatever. So hormones, there's insulin down here. That's a bunch of amino acids. Each one of these is an amino acid. And you have to translate this thing. You just link them together, and we'll talk about that. The ribosome, you link stuff together. Binding proteins are proteins that bind to hormones. But binding proteins are critical regulators in the body too. You send stuff, hormones through the blood bound to binding proteins. So remember IGF, insulin-like growth factor, that has 6 different binding proteins. 1, 2, 3, 4, 5, 6. 98% of your IGF, of your insulin-like growth factor, is bound to those binding proteins. So binding proteins and hormones, we know how these things work, right? We know the importance of these in the body. Mostly it's just a bunch of proteins. Cell surface receptors, these are proteins, right? So what does insulin bind to, or growth hormone, or epinephrine, IGF, all of these hormones, what are these things binding to? Proteins. These receptors are proteins. And if you change the concentration of receptors, or change the sensitivity of receptors, you are altering physiology in powerful ways, whether that's advantageous or disadvantageous. Enzymes. If you want to work in pharmacology, in particular pharmacology, the development of drugs, pharmacy, if you want to be a pharmacist, a lot of this is manipulation of enzymes, and enzymes are just proteins. Enzymes are proteins. A kinase, mTOR, protein kinase B, pKa, PI3K. These are kinases. They attach a phosphate to things. A phosphatase takes a phosphate away, PTEN. That does the opposite of PI3K. It's a phosphatase. Let's convert PIP3 back into PIP2, as opposed to PI3K, PIP2 into PIP3. Phosphoglucomutase, we'll talk about that one a little bit later, a mutase. Let's take a phosphate and just move it. Let's be on the same molecule, but let's reattach it somewhere else, as opposed to attaching or detaching. Let's move it. There's lipases, right? There's proteases. I mean, there are so many different enzymes, and they're all proteins. And when you're getting into metabolic regulation, regulating those enzymes is how a ton of these drugs work. Competitive inhibition, uncompetitive, non-competitive inhibition. What's an NSAID? Non-steroidal anti-inflammatory. What's it inhibiting? COX, cyclooxygenase, right? And so you're inhibiting an enzyme, an ACE. Aspirin is a suicide inhibitor of cyclooxygenase, meaning it's like competitive but irreversible. It's this pretty strong version of inhibition of cyclooxygenase, regulating the function of enzymes, inhibiting enzymes, or augmenting, but usually inhibiting enzymes. Massive regulation over metabolism, and that's a huge percentage of the pharmaceutical industry. Is just enzyme regulation. Structural integrity of tissues, the contractile proteins in muscles, actin and myosin and myomycin, M protein, desmin, alpha actin. There's so many proteins that constitute functional tissue, functional muscle. But all of these have to be made. Every one of these proteins has to be made. If you go get injured or do a really good workout, you're going to have to make a bunch more proteins. It's not just actin and myosin. It's not just contractile proteins. There are all these structural proteins in there as well. Or get into a clot, right? If you're injured in some way, you're going to be bleeding. You're going to be leaking, right? Both blood vessels and lymph vessels, lymphatic vessels. Fibrin and fibronectin, those things cross-link. What are those? Proteins, right? Fibrin is a protein. Fibronectin is a glycoprotein, meaning it has some carb on there too. More protein than carb, but there's some carb in there. So all of these components of these tissues, it's a bunch of proteins. Cellular channels, transporters. Remember that thyroid hormones get into the cell. They get into the cell through transporters. Transporters are proteins. Sodium, potassium have to be pumped against a concentration gradient. That takes a transporter. You have proton pumps, and you have calcium right? Sarcoplasmic reticulum. You got to put calcium back in there against a concentration gradient. All of these cellular channels and transporters, it's a bunch of proteins, right? And then if you get into the immune system, again, I could have put cytokines up here, but that one was already getting too long. But remember, muscle and myokines, muscle releases a ton of cytokines. These proteins, cytokines are proteins, and there's a lot of categories of these things, and they sort of direct cellular responses. They have hormone-like effects. And go work out, and a muscle is going to release a ton of these things, myokines. Some of them are anabolic. If you get into some of the interleukins, 215, stuff like that, this is anabolic. Some of them are catabolic. Some of these, again, some interleukins are also catabolic. But we have, myostatin would be, I guess, the most famous catabolic one of these myokines. But cytokines, it's a bunch of proteins. Cytokines, a bunch of proteins, and then other immune stuff, in the interest of COVID era, not really COVID season, COVID era. The antibodies, right? B cells make these things.
I missed it. You said that cytokines direct. I missed what it was.
Yeah, there's a lot of kinds, chemokines, kind of chemical direction of traffic. Just think cytokine as a small signaling protein that helps direct some sort of cellular response. Immune responses are a big function. Skeletal muscle responses are another function. So cytokines, a small protein that does signaling, and myokines, MYO, would just be like cytokines released from muscle, from myo. Antibodies, let's neutralize some sort of threat, some bacterial threat or whatever. These come from B cells. If the B cells and T cells, the lymphocytes, T cells mature in the thymus, and so they're called T for thymus. B cells mature in bone marrow. So what does B stand for? Bursa fabricius. That's what it stands for, even though it matures in bone. In birds, it should stand for bone. B cells should stand for bone. But in birds, some of these original experiments, that's where they mature in birds. The bursa of fabricius. So bursa is what B cells actually stand for. Even though this doesn't really have to do with us, but you remove that in birds and they don't get antibodies. So the immune system is compromised. But antibodies, these are proteins. Proteins. But we could just do an entire semester on the value, the critical importance of proteins to human functioning, to human survival, to human performance in every sport, in every context, whether it's endurance, whether it is anabolism growth, whether it is some sort of speed and power, something like that. It's proteins are really doing everything. Now, where do proteins come from? Translation. Proteins come from translation, right? This is genetic expression. They come from translation. That's where we're getting all of these proteins. Whether it's mTOR activating this, it's something else. Genetic expression is where all these proteins are coming from. And don't write down the historical stuff, just context for a minute. I'll tell you when to start writing. But Gregor Mendel, this is, if somebody were to say, who is the father of genetics? That's our guy, this Augustinian friar. The Catholic Church has sainted Augustine of Hippo. 4th century born, 5th century died, wrote a bunch of stuff, important figure. He's a patron saint of tons of things. He's a patron saint of theologians, right? That you've made it in life. If you become the patron saint of theologians and like a bunch of cities and whatever, and printers and brewers, he's a patron saint of tons of things. So we get these divisions or sects or almost like fraternities and sororities within religion. This Augustinian, a friar, think like a monk, A monk is self-sufficient. A monk has a smaller dominion. You know, it doesn't have this broader acreage of a province, and a friar is a little bit more integrated with the community and lives on some donations and so. But think of this guy as basically a 19th century monk, and what he was doing was pea experiments, like pea plants, not like urination experiments, but pea plants. Between 1856, 1863, he was working with peas and breeding and testing what was already old wisdom. Nobody had any doubt that there were inheritable traits passed on in lineages. Farmers use this all the time, right? And selective breeding of animals. I mean, everybody already knew that traits were conserved in a lineage, more or less. Not perfectly, but they were retained in lineages. Just like nobody, when Columbus sailed the ocean blue, right? Nobody thought the world was flat. Ancient Greeks understood that the world was round. They had already known the world was round forever. And it doesn't take that much wisdom to be like, oh, look at the horizon. Let me go up a little. Oh, I can see more. The horizon is farther away. Okay, obviously the world's round. Or you can just look at shadows, and yet, everybody thinks that Columbus had something to do with a demonstration of a round world. I mean, it's interesting though that today, my hunch is there are more people who think the world is flat than there was in Columbus's day. There's something fascinating about being in the 21st century and probably having a greater population of people who believe the world is flat than there used to be when Columbus was alive. I don't know the exact numbers, but I would be unsurprised. Now, he was the same thing. He did not come up with the idea of inheritance within lineages. This is just something all farmers knew, and he decided to test it. But a lot of the coinages, a lot of the vernacular is derived from him. Recessive and dominant genes. The terms we still use today come from Gregor Mendel. Now, fast forward the world to the 1930s, everything he did was sort of forgotten for a while until he got to the 20th century. And then it's like, whoa, look how good his stuff is. A lot of famous writers have gone through a similar phase. There were famous artists who don't get famous until after they die. I mean, you look at guys like F. Scott Fitzgerald. His greatest popularity was This Side of Paradise when he was like 23 years old or something, his first ever book. And then he sort of gradually removed himself from the literary map. That's sort of the fate of a lot of today's famous great American novelists. But that was the fate of him too. Like he came up with some great experiments, ran them, documented everything, and then everybody just forgot that he ever existed until the 20th century. But fast forward to the 1930s, I think it was around 1936, Ronald Fisher, the greatest statistician of all time. When people talk about mathematics and we say like, who's the greatest mathematician of all time? Everyone says Einstein, because that's just become a common name. If you say, who is the best writer of all time? Everyone says Shakespeare. Is it true? Not even close. That's not even close to true. But that's what everybody says, and you just have to have a name. You have to have a name. If you're gonna say like, who's the best musician? It's gonna be Bach or Beethoven or whatever. Everybody has to have a name. And if you ask me, which nobody's asking, but if you were to ask me who the greatest mathematician of all time, it's not Einstein, it's Ronald Fisher. This guy, in his head, invented modern statistics. I mean, just like he wanted to grow potatoes better. Like, all right, let me invent, ANOVAs and regressions, and let me modernize the p-value and make it actually usable. Pretty much modern statistics. But he applied modern statistics to Mendel and published, went on record, saying Mendel, everything he did was a falsification and it was an abomination. That's what Ronald Fisher said about Mendel, and I think it was 1936. You can look up the exact quotations and the exact date. But so Mendel, while he was revolutionary to the field of genetics and inheritance, he probably, if Ronald Fisher calls you out, the greatest statistician who's ever lived, calls you out and said, you're an abomination, you falsified your data. I've run it again and again and again, and everything you did is false. You probably cheated. You probably cheated, but you cheated for a good cause. You know, let's become the father of statistics in the process. Now, fast forward the world a couple of decades after Ronald Fisher takes a hatchet to his work. Takes like a sledge hatchet to his work. Takes like a chainsaw to it. And we get the Nature article in which the shape of our DNA, the structure of our DNA is characterized. I wouldn't say discovered because it was sort of stolen, but it was characterized, publicly published for the first time. Now I took, just to show, Watson and Crick, right? This is their paper from Nature. But if you look, Nature 1953, whatever, this is going to come up. I took this screenshot yesterday. I bet if you Google it today, it's 15,980. I bet if you Google it today, there's probably even more citations. This, if there's ever been a quote landmark paper, this is a candidate for like the most landmark paper that's ever been written. 16,000 citations about. So this was Crick and Watson, and they drew it. It's a one-page paper from 1953, a one-page paper. Technically, it exists on 2 pages. You'll see the paper in the file folder. It'll be up later today. But it like starts a little bit into the second page and barely goes on. I mean, the first page is like the initial writing on the first page is someone else's stuff, and then theirs starts a little bit later, and it carries on a little. It's one page, this paper. And it won a Nobel Prize, along with Maurice Wilkins was also put on it. We'll talk about that. This is the diagram they drew, and this is real. I mean, this is 1953. They came up with this thing. This is from the last paragraph in the paper. And again, don't write this stuff down right here, the historical stuff. This is just context. Now, this paragraph goes on just a little bit to talk about, funding sources. There's like one more sentence, but this is the end. We are much indebted to Dr. Jerry Donohue for constant advice and criticism, especially on atomic distances. We have also been stimulated by a knowledge of the general nature of the unpublished experimental ideas of Dr. Maurice, that's what the M is, Wilkins, and Dr. Rosalind, that's what the R is, Franklin, and their co-workers at King's College, London. They stole their work and published it. This is really what happened. And they actually admit it. 1968, this book comes out. It's in my Audible account. It's a great book. It's a great book to a degree. It has limitations. When you watch a Disney movie today, like an old Disney movie, not something that came out in the last 5 years, you watch like a 1979 Disney movie or something, and it begins with that disclaimer about cultural sensitivity. Go to whatever, Disney Plus or something, and they'll begin with, there are elements in this program that were wrong at the time and they're wrong today, and be advised that you're going to encounter this stuff. That disclaimer should be on the double helix. James Watson was a great writer. He was a zoologist from Chicago, great writer. He's talking about Rosalind Franklin. He calls her Rosie. Now, Rosie, Rosalind Franklin, was the only chemist in the 4 people. She was the only chemist. She had a PhD in chemistry, and she was the only one. Watson was a zoologist, and the other 2, Maurice Wilkins and Francis Crick, were physicists, and Crick had a bachelor's degree. Rosie had a PhD in chemistry, and she was the only chemist there. And so Watson talks about her. Rosie, of course, did not directly give us her data. For that matter, no one at King's, the college where she was working, realized they were in our hands. Watson and Crick were given Rosalind Franklin's evidence about the helix structure, and she had written about it previously but hadn't published it. It was in her notes. They were her images. This is her right here. These are the images that they saw, and they didn't know that it was a double helix structure. They did not, this drawing they did after they got a hold, 2 weeks after they got a hold of her data, and she didn't even know. And she wasn't on the publication. And she was left off of the Nobel Prize. And she discovered it. And so it says something about the history in the 50s and again into the 60s. Sexism in science, in chemistry, and in this case, 2 of the 4 were physicists. Crick, who was very smart and wrote a ton of good work, did get his PhD in, I think, 1954 or something after this. But in this article, the Nature article, there's no evidence that Rosalind Franklin, that she knew that in late January 1953, Wilkins had innocently shown her photograph 51 with a stark cross of black reflections to Watson, who was visiting King's. Nor did she know that in February 1953, they were actually given the data, right? That let Watson and Crick see his copy of the Medical Research Council's report summarizing the work of all printed researchers, including Franklin. So she did a bunch of research, hadn't published it yet. Watson and Crick got it without her consent and published. And she was left off of the Nobel Prize. Maurice Wilkins was put on it. Now, why was she left? She died at age 37 of cancer from all those x-rays. She died to discover the double helix structure. And she's the one who discovered it. They didn't discover it. They weren't even chemists. One of them just had a bachelor's degree. She did all the x-rays. She discovered it. It was in her notes, the structure. They got her notes and the images and published. So there's something a little bit sleazy about the whole process. Now, the Nobel Prize, Maurice Wilkins was put on it. The other guy, he was put on it. He didn't really do much other than give the data to Crick and Watson. You can't have more than 3 people on a Nobel Prize. There's no Nobel Prize with 4 people on it, to my knowledge. You can't have more than 3 people. They came up with their 3. They don't give Nobel Prizes to deceased people anymore, but she was alive at the time. And that stipulation, that policy was not in effect. So technically at the time, you couldn't give a Nobel Prize to a deceased person. It's hard to come up with an explanation of how the discoverer of this, they were doing important work too. I'm not taking away from Maurice Wilkins. I'm not taking away from Francis Crick. I'm not taking away from James Watson. We just have to acknowledge that in my estimation, the most important person in all of this was Rosalind Franklin. And she was the one who went without recognition. Now, fast forward the world a little bit more, and really our modern understanding of genetics is the Human Genome Project. The Human Genome Project is really the contemporary understanding of genetics and what genes we have and what those genes do and the function of all of this stuff. This began in 1990, mapping this. And Watson was the director of the Human Genome Project at first for a few years, and then Francis Collins took over. Francis Collins, geneticist / MD, is he's currently the director of the NIH, the National Institutes of Health. That's Francis Collins. He's that guy. He has announced that he's finally retiring at the end of this year. He's apparently retiring. So he has a couple of months left, according to what he said. He's been there for over 10 years, huge figure. He oversaw the Human Genome Project, was Francis Collins. And obviously that's Bill Clinton in the middle. And this is when the rough draft of the Human Genome Project was being announced. This is a rough draft of that. Craig Ventner over here was at the same time figuring out the human genome, privately, not publicly funded. And he was really looking at patenting genes, making a profit on it. He's just a businessman, really smart, very effective, and not as accurate as the Human Genome Project in his technique of doing, of dissecting it all, figuring it all out. But he was fast, he was very fast, and he helped hasten it and contributed to a lot of it. So in the announcement of the Human Genome Project, we have both Craig Ventner, whose company, his own company he was fired from, kind of like the Steve Jobs phenomenon, and then Francis Collins, figuring out the human genome. And the final atlas of this was submitted in 2003, but it continues to change. This continues to change for a lot of reasons. Who has seen the show Parks and Rec? Okay, so you know Ron Swanson. This isn't actually Ron Swanson here, but same actor, Nick Offerman. He's playing Ignatius Riley from Confederacy of Dunces. I point at Marie because she's read it. Confederacy of Dunces, which is one of the most interesting books there is. The guy committed suicide, and his mom took this handwritten pile of sloppy pages to be published, and it won the Pulitzer 20 years later or something. Great book. But so this is him playing Ignatius Riley, who didn't say this anywhere in, I think it was a play. Inside of me is a human genome. It made all of my meat and stuff. Your genome, it says a human genome, which becomes important. I have italicized a human genome. Inside of me is a human genome. A human genome that makes all of my meat, that translates stuff. Now your genome is a bunch of genes. It's a collection of genes. Your genome is a collection of all of your genes. A gene, an individual gene, is a little portion of your DNA. That little portion, the gene, that little portion encodes a protein. This is how you make your proteins. All these sequences, they live in your chromosomes. Chromosomes are in your nucleus. You have 23 pairs of these things, your nuclei, not like one of your nuclei. They live in your nuclei. You have 23 pairs of chromosomes, and your genome is all of your genes. So if you think of your genome like a library, everything, all of your words, a library, it's all of it. Think of chromosomes, shelves or books, something like that in your library, 23 pairs of these things. And then genes, these are individual chapters. Your genes, each chapter tells a little story. Each chapter tells a little piece of a larger narrative. Each gene encodes a protein, which contributes a little bit to your total body, to all of your meat. All of those proteins we were talking about earlier, ion transporters and cell surface receptors and enzymes and like collagen and all of these proteins, all of that stuff. So again, we have 2 copies of every chromosome. Specific genes are in specific chromosomes, and there's about very roughly, about 1,000 genes in each chromosome. Some chromosomes have a lot more, some have fewer. But if you just do the math, it's about, the mean is about 1,000 genes, protein coding genes per chromosome. Again, we have 23 books. Count them, 23, right? Chromosomes. We have 2 pairs of each. We have 2 of each book. We have a pair of each of those. And in each one, again, roughly 1,000 chapters, roughly 1,000 protein coding genes in each one of these things. Now, people will say something like, oh, that's the gene that gives you dementia. That's the gene that gives you breast cancer. That's the gene that gives you whatever it is. People will talk about genetic risk. They'll go to 23andMe and you'll be given this profile of genetic risk. That's sort of an inappropriate way to look at genes, though. We title them accordingly. BRCA genes, BRCA1, BRCA2, BRCA, breast cancer is what it stands for. These genes are named after breast cancer. But think about something like, if I say, what does the liver do? What would your answer be? Use clean?
Releases IGF.
Nice, yeah, releases IGF. It does lots of things. Nobody's gonna say, oh, that's the thing that gives you cirrhosis. That's unlikely to be anyone's answer. Oh, the thing that gives you cirrhosis. I think the liver has additional, and so when we start looking at a lot of these genes, oh, that's the gene that gives you breast cancer. Okay, I think that's sort of a juvenile way of looking at it, at a human genome. But even things like blood pressure, there are a lot of genes that have been identified that contribute to blood pressure. Each one of these genes, each one of these is a chromosome, right? There's your 23 pairs, X and Y, whatever, number 23. But you know, 1, 2, 3, 4, 5, 6, blah, 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 22, and X and Y. The blood pressure gene, the genes that are related to blood pressure regulation, genetic regulation, are all over the place. These things are all over the place. And we get a profile of genetic regulation of blood pressure. But saying, oh, that's the gene that, it's a little bit more complicated than that. There are a lot more players on the field. But see how they're also distributed throughout the chromosomes? It's just not that simple. But the genome really does do a lot of determining, even though I think it's inappropriate to say that's the breast cancer gene. So I think that might be a little bit crude. Your genome, you know, depending on your genes, you might be Peter Dinklage, or you might be Andre the Giant, depending on your, or you could be a, you know, whatever pede, a decapede, whatever, some sort of pede. And so if you, this genetic alphabet, this stuff, A, C, G, and T, and U, we'll talk about later, A, C, G, and T, this genetic alphabet, it really does provide these instructions on how to build the body that you've become. Instructions on how to assemble your bones and muscles and enzymes and brain and all of this stuff. Those genes are the code to build all that stuff. So depending on what that coding says, maybe you're going to be a pede. Maybe you're going to be Andre the Giant. Maybe you're going to be Peter Dinklage. So there is a pretty substantial determinism in the genome, including behavioral stuff, how we respond to a situation. What is our motivation? You know, how determined are we? How lazy are we? All of these things have a genetic link also. So it's not just, oh, the genes are the meat that you have, and then the environment is everything else, including psychology. Nah. Genetics do a lot. There's a lot of determinism. It's fair to talk about determinism with genetics. Now, in the 1960s, we were overestimating the importance of the number of genes, and there was sort of a pride in people saying like, oh, there's like 2 million protein coding genes in the human genome. 1960s, don't write that number down. 1990s, estimated about 100,000 of these protein coding genes inside of us. 2001, getting down under 30,000. Right, today, we still don't know the exact amount today of protein coding genes. And I'll talk about some reasons for that, but grapes have about 34,000. Water flea, maybe 31,000. A fruit fly, maybe 15,000. Ballpark figures for these things. And when you start looking at how we perceive the number of genes, there were cartoons that were coming out about the Human Genome Project. The study found humans have only about twice the number of genes as a fruit fly. Of course, the data could be off a bit. It was done by researchers who had only about twice the number of genes as a fruit fly. What people thought back then, which isn't that long ago, was that the number of genes is somehow important. The number of genes we have is somehow a value. But think about, let's say you're a soccer player. Ryan is a soccer player. Let's say you're Ryan. And like, well, I took 100, shots on goal during the course of that game. I should have won the game. I took 100 shots. Well, did any of them go in the goal? No, but I kicked the ball 100. She would make a lot more goals than this, unless you're a goalie. What is your position? Okay, so I don't know how many goals you make then on defense. I don't know enough about soccer. So we're going to change it back to basketball. If you take 200 free throws, that doesn't mean you're getting any points. It has to actually go in the basket. If you have 1 million protein coding genes, okay, but is it useful? What are those genes actually doing? Are they doing anything of value? If the answer is no, then it's a waste. Okay, you've accumulated 1 billion pennies. Okay, can we get some bills of some sort? Or if you're a baseball player, like, well, I swung the bat 8,000 times today. Well, did you ever hit the ball? No. Well, then you're bad at baseball. The number of genes doesn't actually matter. That said, human beings are somewhere between a chicken and a grape. This is a 2010 study. It's a pretty good one, though, because it also talks about each person has a different number of protein coding genes. Everyone in the room, you probably have a different number. In addition to the ongoing uncertainty about the precise number of protein coding genes, recent evidence has emerged that makes it clear that different humans have slightly different individual gene sets. Most recently, some researchers estimated on the basis of 3 sequenced genomes that gene counts vary by 73 to 87 genes between any 2 individuals. Okay, so it's a human genome. A human genome. It's not the human genome. We don't even have the same numbers of genes. If you've taken kinesiology, functional anatomy, there's muscles in the body, psoas minor, plantaris, palmaris longus. There's muscles in the body that you may or may not have. We don't have the same number of muscles, maybe. The person sitting next to you might have a different number of skeletal muscles. You're almost certainly going to have a different number of protein coding genes. Now again, 2010, there's what the grape was estimated at, a little over 30,000. There's a person, very specific, 22,333. They talk about the limitation. This is our best guess. It's sort of a mean value for a human. And there's a chicken, there's a fruit fly, there's influenza. Influenza doesn't have that many. Now this is 2018, right? This is 8 years later, and there's still uncertainty about the number of protein coding genes. 17 years after the initial publication of the human genome, we still haven't found all of our genes. 2018. All right, the answer turns out to be more complex than anyone had imagined when the Human Genome Project began. Right at the very end of the article. The bottom line is that we don't yet know how many genes we have, although we are making progress. Now, the confidence as we started the Human Genome Project was, when this thing is complete, we will cure cancer, we will cure heart disease, we will cure diabetes, we will cure everything, because we will know how genes behave, we'll be able to intervene, and the solutions will just present themselves. And that didn't happen. The human genome, in 2003, the human genome was more or less fully mapped, and we're a little bit closer to having those things. But again, today, 2021, re-engineering ourselves is not the solution. What the solution is, probably for an awfully long time, is manipulation of our environment, manipulation of our behaviors, nutrition, exercise, environmental stressors. That is how we address disease states. That is how we address athleticism. So, have you seen Quest for the Holy Grail, Monty Python, Quest for the Holy Grail? There's a scene that I have to play, and I hope the volume isn't deafening and piercing.
What makes you think she's a witch? Oh, she turned me into a newt! A newt. Got better.
So newts like salamanders, right? So this salamander, you look up newts or salamanders and DNA, man, they have a lot more genes than we do. All those things are, that and the lungfish also, all these things have so many protein coding genes. And so that would be some remarkable witchcraft if you could take a person who has, low 20,000, something like that, protein coding genes, and turn this person into a newt. Oh, that's impressive. Adding all those genes. Again, the number of genes doesn't matter at all. I mean, this thing outnumbers all of us by a lot, right? But is it doing physics? Is it playing sports? The number doesn't matter. Now, Bill Bryson, he's not a scientist. He's just really what he does is he goes on vacation and writes about his vacations. But he wrote this book called A Short History of Nearly Everything. And it was commentary about the sciences, published in 2003. And one of the lines from that book, chapter 26, is, "Much has been made in recent years of the unraveling of the human genome. In fact, there's no such thing as the human genome. Every human genome is different. Otherwise, we would all be identical. It is the endless recombinations of our genomes, each nearly identical, but not quite, that make us what we are, both as individuals and as a species. Now, if you do have an identical twin, we're talking same genome there, and the environmental differences do become really the dividing factors. Richard Dawkins, in Unweaving the Rainbow, in the opening lines of Unweaving the Rainbow, some of the most beautiful prose ever put to paper was these opening lines. We are going to die, and that makes us the lucky ones. I think this book came out in like 1998, so this is an older book. We are going to die, and that makes us the lucky ones. Most people are never going to die because they are never going to be born. The potential people who could have been here in my place, but who will in fact never see the light of day, outnumber the sand grains of Arabia. Certainly those unborn ghosts include greater poets than Keats, scientists greater than Newton. We know this because the set of possible people allowed by our DNA so massively exceeds the set of actual people. In the teeth of these stupefying odds, it is you and I, in our ordinariness, that are here. Our genes are sufficient to create such diversity. Every single person you have ever encountered, every person they have ever encountered, and every encountered person will ever encounter is a minority of the possible people who will ever exist, catastrophe aside. Now, that's the genotype. The different gene sequences, combinations of genes, that's your genotype. Your genotype is the DNA you were born with. You're conceived and like squirming in a uterus, right? And eventually you emerge and you're growing, and you have a genotype, your genome. It's a four-letter alphabet, with the exception of a fifth letter when we get to RNA. That's A's, C's, G's, and T's. It's a four-letter alphabet. So the adenine, cytosine, guanine, and thymine. 4 letters. And those 4 letters come into these base pairs. There's one there, 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 one there. And they always link up together. Those are the combinations of them linking up. You don't link up thymine with guanine or adenine with cytosine. You don't do that linkage. Those are the linkages that you do. In this double helix, you have 2, they're base pairs, and those pairs are always linked accordingly. And again, this is what it looks like. You have a 5-carbon sugar, and that's phosphate. You know what phosphate is? The phosphorus atom surrounded by some oxygens. Remember, a kinase phosphorylates stuff, attaches a phosphate. That's a phosphate, a little phosphorus and oxygens. 5-carbon sugar, and then there's your A, T, G, C, and you see which one it's linking with. So that's your DNA. That's the structure of your DNA. And you don't need to know the chemical structure of these things, but this is it. This is the 4-letter alphabet. And so you see the base pair, 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 the base pair. These things linking up together, A, C, G, and T. And with those 4 notes, the entire symphony of life, every song has ever been written, the entire symphony of life is 4 notes. A, C, G, T, A, C, G, T, G, T, what I'm telling you, every combination of biological notes. Now here on this little keyboard, I photoshopped this, so there's 5 keys. So if you're in DNA, it's these 3 and one of these. If you're in RNA, it's these 3 and the other one. So we'll talk about that in just a second. So there's sort of 5 keys to it. Uracil is the other one. But in DNA, A, C, G, and T. You say the same thing about, we have this 4-letter alphabet and the entire story of life. Let's get out of a musical analogy and put it in a literary one. The entire story of all biology, not just human life, biology, the entire story of life in the universe as we know it is a 4-letter alphabet that says everything. Now that 4-letter alphabet encodes 20 amino acids, makes proteins out of 20 amino acids in what's called codons, which are nucleotide triplets. We'll talk about that. So each codon, a little section of a gene, encodes an amino acid. It says, link this amino acid here. So we have a 4-letter alphabet that creates a 20-letter alphabet. There are 20 amino acids you're linking into to make proteins. A, C, G, and T are used to tell a story with amino acids. So amino acid, you remember this is insulin right here. So we saw this earlier, that's insulin. Each one of these is amino acid. Just a bunch of amino acids in series. You link them all together and then it folds. It folds in a very specific way. Now, we use a 4-letter alphabet to create a 20-letter alphabet to create an alphabet of proteins. And how many proteins are there? I don't know. At this point, you can just say, who cares? It's not that who cares, it's just that who knows. At some point in the future, who cares and who knows will be solved by much smarter people. But this paper right here, the number of different proteins comprising the human proteome is a core proteomics issue. Researchers propose numbers between 10,000 and several billion. Okay, so at this point, who cares? But what we do is we have a 4-letter alphabet, A, C, G, and T, which creates a 20-letter alphabet of all those amino acids, which creates an enormous alphabet of proteins, which create us, which create our phenotypes. So the genotype creates our phenotype. Your genes determine whether you're an armadillo or a fox or an ant or a panda bear or a polar bear or a regular bear, whatever it is. Your genome creates your phenotype. Now for people, your phenotype is different, right? For people, maybe you're a banana-shaped body, or a potato-shaped body, or a pear-shaped body, or an apple-shaped body, or from Mike Tyson's Punch Out, Super Macho Man, it's like an upside-down pear, right? I guess he's apple or something, and he's maybe potato, and he's like upside-down pear. So our different physiques, our phenotypes, are in part determined by our genes, in part, because environmentally, behaviorally, you can control those things too. Now, it goes farther, and we're going back to Richard Dawkins again. He wrote a book called The Extended Phenotype. Ants make, I don't know, farms? What do ants make? What is like a little ant house? Ant hills. Ants make hills. Beavers make dams. Birds make nests. Spiders make webs. People make like pants and cars and stuff. People make weird stuff relative to what the rest of nature makes. Bees make hives. Bees don't make dams, and beavers don't make hives. The extended phenotype is we have A, C, G, and T creating an alphabet of amino acids, creating an alphabet of proteins, creating an alphabet of creatures who create the world as we see it. The extended phenotype is if you have a spider's genome, you are going to make webs. Webs is part of your phenotype. If you are a beaver, you are going to make dams. The dam is part of your phenotype. And if you're a person, I don't know what you're making, your art and music and baseball bats or whatever. The extended phenotype, it really is part of us, but we create stuff out of the tools that we have. So your phenotype is not the exact same thing as your genotype. There are differences, and you can think of your phenotype as your genotype with some highlighting in it, some genes being expressed, a bunch of mTOR signaling or something, and a bunch of deleted scenes. You're chopping stuff, you're cutting stuff, they're deleted scenes. there are genes that aren't being expressed. Genes that you are not expressing are sort of deleted scenes. Yeah, you filmed it, it's there, right? It's in the studio, it's at the back of the DVD, it's whatever. It's there, but you don't release it, the unreleased stuff. That is genes that aren't expressed. And so a tail in a human being, there's tons of data on not tons of people, it's rare, but there are lots of studies talking about human tails. Now, we don't express those genes. Occasionally, somebody who had the genetic misfortune to express those genes rarely can grow a tail. But we have these relics of ancestry in our genome, and it's a deleted scene. We don't express those things, you know, but once in a while, somebody will. Once in a while, you'll get a little tail. Atavism, this is these sort of relics that you can, it turns out you're capable of expressing. But we have genes we don't express. We have genes we do express. Among the genes that are active, that we are expressing, that aren't just dormant, lying there sort of useless. Among the genes that are active, we express in varying amounts. And so a phenotype is the expression of your genes. And how do you change gene expression? You know, how do these genes make you a cherry or a banana or whatever? How do we get these different figures? Oh, go lift weights, right? If you've seen The Office, when Dwight Schrute had his gym, everybody who goes in there is changing genetic expression. If you go for a hike, get up to altitude, right? Eat a bunch or fast, drink a bunch or become very parched, sleep a bunch or be sleep deprived. All of these things are changing genetic expression. They're changing the behavior of your genes. If you simply eat a banana, you're going to get an insulin response. That insulin binds to its receptor. Downstream from that, you're getting translation differences. You're getting genetic expression changes. Everything you do affects the behavior of your genes. And you know, for AMPK, at the end, we're seeing translation inhibition. mTOR, at the consequence, the downstream consequence of mTOR is translation. And we know there's a lot of stuff that can turn that on, and there's a lot of stuff that can turn it off. So we have our DNA, we have our genotype, our DNA, we get transcription, this is in the nucleus, transcription to get RNA. RNA is translated at the ribosome to link together a bunch of amino acids. So we link together a bunch of amino acids in order, very specific order, and that's how we get proteins. Proteins fold. You know those magnetic, like bead necklaces? Where you have the like little metallic, it looks like a necklace of little metallic beads, and like you put it down on a table and it all folds up. That's sort of what these proteins will do. You link together all these amino acids, and then they fold up, but they fold up very specifically. They can be misfolded, but they fold up very specifically. So DNA, this is your genotype, gets transcribed. You get transcription to get your RNA, which gets translated at the ribosome, linking together amino acids into a protein. Protein folds, and you know what all these proteins do. That list of things we talked about at the beginning. You know, transporters and hormones and binding proteins and myokines and all that stuff. So step one, transcription. This is in the nucleus. Transcription, nucleus. RNA leaves the nucleus, goes to the ribosome. At the ribosome, it's translated. That mRNA gets translated into this amino acid chain, and then the chain folds into a protein. So each gene is a bunch of this A, C, G, and T, but for the RNA, you're going to get a replacement with U's. You're gonna get a bunch of U's in there. Same thing though, it's the same recipe. And so the DNA becomes RNA, transcription, goes to the ribosome. There's two subunits. There's a reader and really a linkage subunit. And that's how we get our proteins. So there's the subunits, right? The small subunit, that's the reader. And then the large subunit is doing the linkage. It's linking together those amino acids to create your protein. So you're reading the RNA. Down here, you see a codon right here, and another codon, and another codon, and another codon, and another codon. This is your RNA. And its nucleotide triplets are codons. And so, again, we have this A, G, T, C, and then that 5-carbon sugar right there, and then the phosphates. This is the DNA, the A, C, G, and T. And so the base pairs would be these, base pairs are those, base pairs are those, base pairs are those. And these code for protein. So you have your first codon, which codes for methionine. The first one is always going to code for that. Your start codon. And then you have a stop codon at the very end. A start codon initiates translation. A stop codon says we're done here. We have translated a protein. Go be free, protein, go behave, do what you were created for. So nucleotide triplets, each one of those triplets codes for an individual amino acid. And again, RNA has uracil. DNA has thymine. So these two, the A, C, and G are in both. Uracil is RNA only. It replaces the T. U replaces T. Other than that, the recipe is the exact same. We'll go over this again next lecture, but again, there's a start codon. You see methionine right here, start, and there's a few potential stop codons. UGA, stop codon, UAA, stop codon, UAG, stop codon. That says translation is over. We have created our protein. Once you hit, in this case, UAG, once you hit that one, UAG, stop. AUG, start, wherever it is there. AUG, start, methionine. So the sequence of amino acids just really matters what these 2, 3, 4, 5 are. The sequence right here of these nucleotides, ACG, GAG, CUU, CGG, AGC. Each one of those codes for an individual amino acid, and the sequence of amino acids determines what protein it is. Did we just make insulin? Did we just make IGF-1? Did we just make Titin? 30,000 amino acids is a big one. That one takes a while to translate. So there's a start codon, which initiates everything. There are intermediate codons, which each add an individual amino acid. And there's a stop codon that ends the whole process. And the reason these codons are so important is because proteins do everything in your body. Right? The hormones and binding proteins and cell surface receptors and enzymes and the structural integrity of tissues. All of these proteins, you have to translate them. And there are differences. There are differences in, not here, if they're identical twins, but from person to person, there are differences in our genomes. And this will be the last slide we talk about. This is a diagram I did for this article. It's Garrett Ash. We did our PhDs together. But that blood pressure one, these are all of these identified blood pressure genes. And so our genes are stored in chromosomes, right? We get transcription to RNA, translation, and folding into proteins. Proteins become these biological networks. Those biological networks have clinical importance, clinical importance, how we respond to exercise, our motivation to exercise, our need to exercise. Okay, that's it. The important stuff we'll talk about again next lecture, and the history I'm going to leave off. But what we'll talk about, transcription, translation, these codons, how all this stuff works. And then we'll get into some aging and deterioration and moving into geriatric folks.